after narrating the lost and found story of Mendel and narrating the belated case of recognition of the great scientist, let us now pass on to another aspect of Mendelian genetics, and that is the methodology of Mendel. And before going to the laws and their interpretations, even in the modern sense, one has to be very clear about the terminology that was used by Mendel and the modern terminology which metamorphosed thereafter. As you can see on your screens, the flower of Pisum sativum is a good looking one with a papilionaceous corolla, and then one has to only remove the keel petal because it's a cleistogamous flower and it will be open, and one can manipulate the stamens, one can remove the anthers, and one can dust a foreign pollen grain onto the stigma and then move the keel back and even cover the entire flower with an envelope or a water to be isolating itself from the other foreign pollen grains. So, the experimental methodology that Mendel employed was, first he selected the plants of his choice with the desired characters, and then he performed hybridization. As I just said before, hybridization included opening of one of the flowers and surgically removing with the help of a razor the stamens. So, technically this flower now became a female one. Then the pollen grains from the other parent with the desirable contrasting character was dusted onto it, and the keel was then set free, and finally the flower was bagged. And then he waited for fertilization to take place, and he collected the seeds. He then again the seeds were sown and a new generation of plants were got to find out as to what happened to the pattern of inheritance of those characters. He also performed selfing experiments, and very naturally, because pea plant is a cleistogamous one, he selected 7 characters, and these characters, they ranged from the seed shape, seed color, then the color of the flower, the nature of the fruit, the shape of the fruit, that is the color of the fruit, the position of the flower on the axis, and so on. These 7 characters, somehow one fails to understand why he collected 7 characters, because at that time there were no idea of chromosomes. As fate would see, the Mendelian genes may not be present on all these 7 chromosomes, because chromosomes in Pisum sativum 2n is equal to 14 means that the haploid set of chromosomes in garden pea is 7, and it was much later that it was found that the 7 characters which were chosen by Mendel actually resided on 4 chromosomes, namely chromosome number 1, number 4, number 5 and number 7. If one looks very critically but briefly at this slide, you will find that on chromosome number 4 there are 3 characters, or now we can call the term genes for the 3 characters, and out of them 2, that is the pod shape and the plant height, they are too close to each other. So, when Mendel made a cross between the ones which I am mentioning to you, the pod shape and the plant height, results definitely were at variance. He did not understand, and he did not report this cross in his findings at all. Later people came to know that this was a case of linkage. We will have ample opportunity to talk about linkage, so we will again come back to this very diagram. The point is that one should now be sure and familiar also with the terminology of Mendelism before we embark upon studying Mendelian principles, the laws. Even before doing that, let me just remind you about a misconception. People believe that Mendel's laws were given by Mendel. No, Mendel's laws were formulated by Karl Correns. Incidentally, Karl Correns was a student of Nageli. One will have to argue whether it was a case of mere coincidence or as destiny would have it. Erich von Schermach, who was again one of the rediscoverers of Mendelism, his maternal grandfather, Leopold Frenzy, was Mendel's teacher at the University of Vienna, under whom the whole knowledge of botany was imparted to Mendel. But the laws that were made were two. So that means there are two Mendel's laws given by Karl Correns, of course based on Mendelian principles, the first being the law of segregation and the second law of independent assortment. Hugo de Vries, the great Dutch geneticist, who had also talked about, who gave the term mutations, had also reached very similar conclusions, and he had also called as the principle of the purity of gametes. So the terminology sometimes get confused as to who gave which term. Let's begin with the concept of a gene, because this term has undergone so much of metamorphosis in less than 6 decades. The term gene was given by Johansen, obviously much later than Mendel's rediscovery and Bateson's appreciation of Mendelian laws, because Bateson had also reproduced and replicated all the principles of Mendel in animals and found them to be absolutely true. So a gene would be defined as the smallest structural and functional unit of heredity, and gene term has come from the Greek word genos, which means taking birth. Mendel never mentioned the term gene, but then in his writings he refers such particles as Anlage, and he also talked about a word factor, which was also picked up by Karl Correns, who himself introduced the term elemente. So that means, by that account, Anlage, factor and elemente, they should all be considered the precursors for the term gene. The concept of a gene has undergone a lot of transformation, as I said before. It was in 1941 when two scientists, Beadle and Tatum, working on Neurospora crassa, found out that it was one gene which was responsible for the synthesis or controlling the synthesis of one enzyme, and this gave birth to one gene, one enzyme hypothesis. Still, it was felt that the gene was indivisible, and the accent was as to what really it was controlling. As enzymology grew further, Janowski and Ingram in 1950s found out that a gene may not be controlling one enzyme altogether all the time, because many of the proteins would be made up of more than one polypeptides, hemoglobin being the classical example, and a change in just one of the amino acids in one of the alpha or the beta units could do the trick. So they modified this idea into one gene, one polypeptide hypothesis. So at least the enzyme was now divisible, but then the gene was still untouched. After some time, when molecular biology was at its peak, In the 1960s, Semyor Benza was the first person to study the gene in details, the functional aspects, and he said that the gene can be further divisible into cistrons, that is the functional units, the recons, the recombination units, and the mutons. So, by that analogy, one gene could be composed of either one or even more than one cistrons, and on the basis of which came the concept of monocistronic and polycistronic genes. So, this needed an overhauling of the initial idea, and now the dictum became one cistron, one polypeptide. This is the current idea, which is of course open to further clarifications and modifications. Then comes the concept of a character, because we have been talking all this while. He chose 7 characters, what were the traits in Pisum sativum, and so on. So, how do we define a character? A character is a well defined morphological or physiological feature of an individual. You say Mr. So and so has blue eyes, Mr. So and so is very tall. So, you are talking about eye color or height. Mind you, one should not get confused in character and trait, because some people believe they are synonyms, but let us see how do we distinguish. The character is eye color. We do not mention what type of color, but we say it is a character eye color, and height similarly is a character. Then what would be a trait? A trait would be a characteristic distinguishing feature of that particular character. We say what type of eyes, then the trait is dark or they are light. What do we mean by height? An individual or a plant is tall or dwarf. The other character would be color of the flowers. The trait of this character would be either red or white, and so on. So, that means it is these traits which are heritable and they will pass from one generation to the other. In this context, let us view the diagram which we had seen before, referring to Mendel's 7 characters of choice, to make the whole discussion more understandable. Mendel never took 7 traits. He chose 7 characters, which means 2 of the characters were from seed, namely seed form, seed nature of the cotyledons, then the nature of the flower, that and the nature of the pod and the plant. So, what were the traits that he took? For example, in the seed form, the traits were contrasting. As we said before, sharply contrasting. The seeds were either smooth textured or they were wrinkled. So, these are the 2 traits which make up a character. So, in that hierarchy, character would be a higher one. Then comes the concept of alleles, which has created a lot of confusion. One talks about a gene and allele as a synonym many a times. The term allele was given by William Bateson himself in 1902, which says that they are different versions of a gene. What do we mean by different versions? That means they constitute one or more alternative forms of a gene, but then the prerequisite is that they should be positioned, they should be present at the same locus on a pair of homologous chromosomes. Here we introduce 2 more terms, a locus and the homologous chromosomes, which we need to define. So, that means usually we said the one or more, that means usually there are 2 alleles if there are alternatives to a gene, and their symbols need to be italicized. So, that means if we are saying this versus that, top versus bottom, so that means there are only 2 alternatives. However, when we come to a study of blood groups, then we get a bit more of this information, as saying that sometimes the alternative forms of a particular gene could even exceed. There would be more than 2, especially in case of multiple genes. And also, when we talk of quantitative inheritance, we would not say simply as this or that. We would say this also, that also, and that. So that means there would be various intermediate forms. So then first let us define what is a locus. A locus where the alleles are present, that means it's a specific place on a chromosome where a gene is located. So that means on a set of chromosomes the position is predetermined. For example, in the picture that you are looking at, the homologous chromosomes, if this position on one of the chromosomes is of eye color, then the corresponding position on the homologous chromosome has to be of the eye color. Whether the eye color is same or not, that is immaterial, but then it is only of eye color and eye color alone, not even the texture of eyes. So that means they occupy the same locus. And what do we mean by a homologous chromosome? Each of the chromosomes which are present in a diploid organism, they are coming from one parent, each one of them, and they are going to form a pair or a counterpart. In other words, each chromosome coming from a parent has its counterpart coming from the other parent. For example, Pisum sativum has 14 number of chromosomes in its diploid complement. So that means 7 are coming from one parent and 7 from the others. So it makes what is called as 7 homologous chromosomes. If we look in a little more details about a homologous chromosome, which seems to be a submetacentric chromosome, so we find that the loci, the plural for locus would be loci, or the positioning of a particular gene, say for example of the petal color, they would occupy the same position or the same locus on a pair of homologous chromosomes. So in other words, allele can be considered as a version of a particular gene. An alternative form of a gene would be an allele. In this diagram, we've shown one allele to be written as capital R and the other as a small r. We'll just come to that. The homologous chromosomes will undergo meiosis. First, prior to meiosis, they have to duplicate, and the duplicated version would mean that there are two identical copies of every chromosome, which would mean that they are having sister chromatids. The crossing over would take place between the non-sister chromatids of a pair of homologous chromosomes at the Pectine stage of meiotic prophase 1. So that means each allele is going to control different expression of traits of the same character. Coming back to the confusion between the character and the trait, it means, for example, if there is an allele R, say we put it as a capital 1, it is controlling red color or purple color, and an allele small r in the lower case is controlling another trait which is the white flower. These are two different traits, but the character is the same. And what is the character? The character is color of the flower. So that means, how can a gene therefore express itself as both the alternatives of the character at the same time? You cannot be on the ground floor and top floor at the same time. Either you will be at the end or you will be at the beginning. So in other words, one of these characters would be expressed. This was one of the basic axioms of Mendelian principles also. Now, what do we mean by the terms genotype and the phenotype? These terms incidentally were also coined by Johansen in the year 1911. Johansen was the scientist who was also very ardent supporter of Mendel. He said that what is the external appearance of a character? We see a plant having a flower color, a person who is bald, a person who is tall, a person who is short, a plant which has red flowers or white flowers. So that means we are only describing the external appearance of a character, that is what is the flower color and what is the texture of the head and so on. So this is the phenotype. The genotype, a phenotype would change perhaps with the change in the environmental conditions. If you were to expose yourself to sun for a very long time, perhaps you will be tanned and the complexion may change. The genotype again would be the genetic constitution of an individual, but then you have to express it as expressed by using these symbols. Let us look at this very carefully. Capital T or capital T small t, or both the T's in a lower case, or T nothing. Here nothing is denoted by a cross, or small t nothing, or nothing and nothing. Why have we expressed it in pairs? Because this is what the genetic constitution would be on a pair of homologous chromosomes. We should therefore remember the genotype of an organism is always expressed by the symbols which are italicized. Now, depending upon again these symbols, Bateson and Saunders, they gave the terms homozygous. Homozygous, we are always talking about an organism which is diploid, because you will agree with me that a haploid can never have alternatives. If I just give a wheel in your hand, I mean, you really don't know whether it is the front wheel or the rear one. I have to give you two to get to that conclusion. So that means the entire discussion is revolving around a diploid organism. So a homozygous constitution would be a diploid organism where both the alleles are of the same type. Either both of them are capital or both of them are in the lower case or the small letters. So that means in this case, if meiosis is going to take place, both of them are capital B and capital B, or small b and small b, naturally they will produce only one type of gamete with reference to the locus. So that means you can't call them as a true breeding individual. So these are the true breeding or the homozygous organisms. On the other hand, heterozygous organisms are ones where there are two different alleles which are present at a specific gene locus. This would be called as impure because both of them are different, or a hybrid. How would you depict them graphically? Say for example, capital T and small t. Depending upon what we are hinting at in terms of a character, so we are taking an abbreviation, capital B or small b, capital S or small s. But when meiosis occurs, then an impure or what we call as a hybrid or a heterozygous individual will produce two types of gametes, one capital T and one small t, one capital B and small b. So that means this is a case where two types of gametes are there. On the picture, on the screen, you could see a homozygous individual which is capital B and small b, whereas the heterozygous ones are the capital B small b, or it could be the other way around, small b and capital B. There is another very interesting situation, and which is hemizygous. What would happen if there is just one chromosome and there is no allele on the corresponding locus, or for that matter there is no corresponding or homologous chromosome at all. So that means a diploid organism which has only one of the two alleles present at the specific gene locus would be a hemizygous one, because the other allele is simply missing. So we can depict it as AX, small a and X, that means only capital A is present or small a is present, or for that matter like what we have in our genetic constitution, one X and one Y as far as a human male is concerned. So if there is one X and one Y, then there is no corresponding allele to interact, and therefore X will go its own way and Y its own. So that means these are the basic ideas of homozygous and the heterozygous. Maybe in some of our next episode of genetics, we talk about the dominant, the recessive, the codominant of the ideas, because whenever we talk about two of these alleles, and I said that both of them cannot express themselves and there is no blending between them, otherwise the results would have been intermediate, one of them needs to be dominating. So this is what exactly a dominant trait is. That is, a dominant trait will always express itself whether it is a homozygous condition, a heterozygous condition or a hemizygous condition. So anything which dominates, we would like to keep it as a capital letter. So for that matter, capital B or capital B small b, this is a heterozygous one or a hemizygous one, right. We will talk about the modern notations of dominant traits on the next turn. Thank you.